All right, so we are going to spend the next 3 weeks talking about the nervous system. All right, so this week we're going to talk about neurons and electrical signaling, then we'll talk about the central nervous system, and then the peripheral nervous system. All right, so today we're going to do a brief overview of the nervous system because we're spending 3 weeks talking about it. Right? And then we're going to talk about the resting membrane potential again. Because one of the great things about neurons is that they can change their membrane potential. So everyone needs to understand resting membrane potential before you can understand how neurons change their membrane potential. So I told you need to understand membrane potentials for exam 1, and then obviously for exam 2 as well. And then they'll come into play with exam 3 as well. Okay? So take the time to learn it. We'll talk about graded potentials versus action potentials. We'll talk about propagation of the action potential and then the synapse. So all this information is in chapters 7 through 8. You might notice we've just skipped chapters 5 and 6. We'll come back to them. Okay, so we're going to cover the endocrine system later in the semester. Okay, I find that everyone's had it with chemistry by this point, and I prefer to put nervous system all in one exam, so that's why we've skipped chapters 5 and 6. Don't worry, we'll go back. Okay, so communication is critical for any relationship as well as for homeostasis, okay? So your cells need to communicate with one another, and we have 2 body systems where their key role is communication. And we're going to talk about the nervous system. The nervous system communicates rapidly. Okay, so the nervous system does rapid communication. The endocrine system also plays a key role in communication, right? But it can do communication over longer periods of time. So it controls things like growth and reproduction. So body processes that can occur over a longer period of time, whereas the nervous system controls what has to happen now. Right, the nervous system is made up of nerves. Nerves are bundles of axons. Okay, so nerves are not cells, right? They're bundles of cells. They're bundles of neurons, right? And it's the neurons that undergo rapid changes in their membrane potential. Okay, and that acts as the signal. Okay, so neurons change their membrane potential. It goes from minus 70 millivolts all the way up to positive 30 millivolts. And that acts as an electrical signal, super rapid. Okay, so here we have the overview of the nervous system that we're going to talk about for the next 3 weeks. So in this brown box is the central nervous system. And this is where your brain and spinal cord reside. It's the command center. Okay, it's receiving information from the afferent branch of the peripheral nervous system. So everything in yellow is the peripheral nervous system. So the afferent branch sends signals to the brain and spinal cord. And those signals are your somatic senses, so that's sensations coming from your skin surface. Okay, your special senses, so that's vision, smell, taste, hearing. Okay, and then your visceral senses. So that's information coming from your visceral organs. Is your stomach filled with food or is it empty? What's the heart rate right now? Okay, you are not consciously aware of your visceral senses. Right? You are consciously aware of your somatic and your special senses. Okay, so the peripheral nervous system affects the central nervous system. So those of you who have a problem with affect versus effect, you can learn it through physiology. Okay, the afferent branch sends information or affects the brain. Okay, the brain and the spinal cord are the command center. They're going to integrate all that sensory information, and then they're going to send out signals via the efferent branch. So the efferent branch carries the effects of the central nervous system. Okay, so the efferent branch can be broken into the somatic and the autonomic Somatic controls your skeletal muscle. This you can control consciously. Okay, you have conscious control over the somatic branch of your peripheral nervous system. Okay, the autonomic is your sympathetic and your parasympathetic, which controls cardiac muscle, smooth muscle, glands. Enteric nervous system is what controls your GI tract. Okay, do you have conscious control over these tissues and organs? No. Okay, so autonomic means it happens automatically, okay, without your conscious control. Parasympathetic is rest and digest. Sympathetic is fight or flight. We'll talk about afferent branch and efferent branch after we talk about the central nervous system. But today, we're just talking about neurons. Okay, so our nervous system, we've got neurons, which are the excitable cells. Okay, they're the ones that can change their resting membrane potential. But they're divas. They need loads of support cells. And those support cells are called glial cells, right? So the glial cells are all the support cells. So here we have a interneuron in the brain. This is being shown in yellow. Okay, we have some epidermal cells. They help make and circulate cerebral spinal fluid. Okay, we have some oligodendrocytes, which myelinate interneurons. In the peripheral nervous system, Schwann cells do the myelination. Right, we have some microglia, which act like immune cells within the central nervous system. Oh, and then in blue here, we have the astrocytes. And notice the astrocytes have these foot processes that are touching everything, one of which is the capillary. It causes the formation of the blood-brain barrier, makes those capillaries form tight junctions. Okay, and then it also helps guide neurons during development, also helps take up excess potassium and neurotransmitters. Astrocytes play a big role in the central nervous system. We'll talk in more detail about these glial cells when we talk about the central nervous system. But just so you know, we're going to talk about neurons, but there's lots of other cells in the nervous system. Okay, the neurons need loads of support. Okay, so let's do the anatomy of a neuron. Okay, so that we're all on the same page and we can talk about action potentials because we can talk about specific parts of the neuron. So here we have some neurons and we have dendrites. Okay, dendrites are going to receive signals. Okay, notice we've got these little green arrows. So they're going to have synapses with maybe neurons that are presynaptic to these or upstream. Okay, and that's going to have a release of neurotransmitter. That dendrite's going to have a receptor for that specific neurotransmitter. That's going to alter membrane potential. Then the dendrites here are attached to the cell body. Okay, the cell body contains the nucleus. It's also known as the soma. It's where protein synthesis is going to occur and neurotransmitter synthesis is going to occur. And then we have what's called the axon hillock. The axon hillock is the start of the axon. Okay, the axon hillock is where you get signal integration. Okay, so this neuron is going to be receiving information from a whole bunch of presynaptic neurons. Okay, and then all those signals are going to integrate at the axon hillock. And the axon hillock is the first place an action potential gets fired if one is going to be fired. Okay, so the axon hillock determines whether or not that neuron is going to fire an action potential. And if the axon hillock fires an action potential, that action potential is going to go all the way down the axon to the axon terminals. Okay, and those are just the ends of the axon. And this is showing that this presynaptic neuron is only synapsing on a second neuron, but oftentimes these axon terminals are going to be synapsing on other neurons. Okay, but this, for simplification, is showing that all the synapses are on this postsynaptic neuron. Okay, presynaptic neuron means the neuron before the synapse. Postsynaptic neuron means neuron after the synapse. Right? So that means this presynaptic neuron, if we had a third neuron up here would also be a postsynaptic neuron as well. Okay. So let's look up at the blow up of the synapse. So here we have the axon terminal of the presynaptic neuron. It releases neurotransmitter, and then that postsynaptic cell has to have receptors for that neurotransmitter. Okay? Notice that we've got synapses occurring on the dendrite as well as on the soma. Okay, so synapses don't only occur on the dendrites, they can also occur on the somas. Here would be the axon hillock again. Here's the axon, and here we're seeing a collateral axon. That's going to synapse on a different neuron altogether, right? So the axons can branch. Okay, so here in words, we have the major components of a neuron you need to know and their roles. Okay, so the soma is the cell body. It's where the nucleus and organelles are. Okay, dendrites receive incoming information. Axons are transmitting that action potential. Axon hillock is signal integration. It's where action potentials are initiated if they're going to occur. Okay, axon terminal releases neurotransmitter, and synapse is the space between the axon terminal of the presynaptic neuron and the dendrite or the soma of the postsynaptic neuron. Okay? And that space is just filled with extracellular fluid. If that synapse occurs in the central nervous system, that extracellular fluid is cerebrospinal fluid. Okay? If it occurs in the peripheral nervous system, it's just interstitial fluid. Okay? This means that all neurotransmitters are water soluble because they have to diffuse through extracellular fluid. Right. There are different styles of neurons. We have afferent neurons, we have interneurons, and we have efferent neurons. Okay, so this figure looks a lot like the one where we look at the organization of the nervous system, but here we just have neurons being shown. So here's the peripheral nervous system, here's the central nervous system again. The neurons that reside completely within the central nervous system. Okay, they don't have axon terminals that leave, they don't have cell bodies outside of it, etc. All of the neuron, dendrite, soma, axon terminals are within the central nervous system. They are called interneurons. Okay. The peripheral nervous system has afferent neurons and efferent neurons. Afferent neurons send signals towards the central nervous system. Efferent neurons send signals away. Okay, so the afferent neuron, notice we've got the cell body here, and we have dendrites way over here. For afferent neurons, the dendrites do not occur on the cell body. Okay, the dendrites are often specialized endings that are some sort of sensory or visceral receptor. Okay, so they're going to be detecting that sensory information. It might be a pressure receptor, so it detects when you've touched your hand. Okay, sometimes they're separate cells, so photoreceptors in your eyes are separate cells that then send a signal to an afferent neuron. Right? And then we've got the axon hillock right here. So where all the dendrites converge is where you get the axon hillock. And then the axon. Okay, so the cell body does not occur with the dendrites. Right? And it's actually going to occur in a ganglion. Okay, so where the cell body occurs is called a ganglion. And a ganglion is any time you have a cell body outside of the central nervous system. A neuron cell body, obviously. Yeah?
On the components of a neuron slide,
Yeah.
I thought that the axon hillock was right where the axon meets the body.
Not in this case,
Oh, okay.
right? That's why I'm pointing it out. Not in this case.
This is the difference.
Yep, so this is the difference. On afferent neurons, the axon hillock occurs where all the dendrites converge. Okay? Not at the cell body. For efferent neurons, you got the dendrites in the cell body occurring within the central nervous system, right? And then the axon runs into the peripheral nervous system, And then it's going to synapse with an effector organ. And the effector organs of the efferent peripheral nervous system are skeletal muscle, smooth muscle, cardiac muscle, or glands. Right. Any questions about neuron anatomy? That's the really important stuff you need to know. We're talking about action potentials. All right. Let's talk about, where?
Right here.
Oh.
So, as far as the axon hillock goes,
Yeah.
You said that it doesn't have to be touching the cell body when it's on the ganglion, right?
Right.
So is that where the action potential starts as well?
Yes, the axon hillock is where the action potential starts.
Okay.
No matter if it's at the base of the cell body or the base of the dendrites.
Okay.
Yep. So the axon hillock is where the action potential is always initiated. Okay. Any other questions? Okay. Let's talk about the resting membrane potential again. Okay. So for most cells, the resting membrane potential is minus 70 millivolts. Okay, so cells have a resting membrane potential of minus 70 millivolts. This is for non-excitable cells as well. Okay, we're going to talk about neurons, which are excitable cells, but all of your cells have a resting membrane potential where the interior of the cell is more negative than the exterior. And the interior is more negative because of negative charges that are trapped inside those cells. And those things that are negatively charged and trapped inside of your cells are your proteins. Excellent. Okay, and then we have more positive charges outside the cell. And the major extracellular cation is sodium. Excellent. Okay, so there are two factors that are critical to determine resting membrane potential. Ion, electrochemical gradients. Okay. And how permeable the membrane is to these ions. Okay, so whether or not there are leak channels. And which ion do our cells have leak channels for? Especially a lot of them. Potassium. Exactly. Our cells are permeable to potassium at rest because of those leak channels that are always open. Okay, so we're going to draw our cells. Go ahead and make a circle. Put positive on the outside, negative charges on the inside. Put your intracellular anions that are trapped on the interior of your cell. Those are your proteins. Why are proteins negatively charged? What's their functional group that gives them their negative charge? Carboxyl groups. Remember those carboxyl groups give off the hydrogen ion? Okay, put in your sodium-potassium pump. The sodium-potassium pump is a primary active transporter or a secondary active transporter? Primary, because it directly uses ATP to pump 3 sodium out, 2 potassium in. Okay, so it's setting up the sodium gradient and the potassium gradient. It's using one molecule of ATP to pump 3 sodium out, 2 potassium in. Okay, so that sets up our sodium gradient where you have lots of sodium outside, very little inside, and our potassium gradient. Lots of potassium inside, very little outside. It's all due to the sodium-potassium pump. Okay, let's add our chemical versus our electrical gradient. So chemically, sodium wants in, right, down its concentration gradient, and electrically it wants in as well. Right, chemically potassium wants to leave, but electrically it wants to stay inside the cell, but which is stronger? The chemical. Okay, so if it has the chance, potassium will slowly leave the cell. Okay, let's add chloride. It's sodium's best friend, so it's high concentration outside, low inside. Okay, and so chemically it wants into the cell, down its concentration gradient, but electrically it's being repulsed by those anions inside the cell. Okay, so it also has opposing electron chemical gradients, just like potassium, but which one is stronger? The chemical. So if given the chance, chloride will enter the cell. Okay, and then lastly, we'll add calcium. So calcium is in higher concentration outside the cell than inside, and I made the calcium smaller than the sodium, a smaller font, because remember there's way more sodium than calcium. Okay, you just have way more sodium in extracellular fluid than you have calcium. Sodium is the major extracellular cation. And why does calcium get a 2+? Yeah, in the back.
Because it gave up 2 electrons, not just 1.
Exactly, it gave up 2 electrons, so it gets 2 positive charges. Okay. Okay, chemically it wants to enter, and electrically. So it also, just like sodium, really wants inside the cell. Oh, yeah.
Before you've drawn some X's, some zeros, what do those mean again?
Oh, so when I draw the X's, I mean that cells aren't permeable to those ions, right? So we drew X's through all of them except for potassium because of those leak channels. Okay. Now we're going to think about what would happen to membrane potential if the cell became permeable to this ion or this ion. Okay, so here we have resting membrane potential, and you can think of this as a y-axis on a figure. Okay, so we've got resting membrane potential at minus 70 millivolts, and resting membrane potential is V subscript M. That's how it's denoted. Okay, and then we have 2 equilibrium potentials on this y-axis. We have the equilibrium potential for potassium and the equilibrium potential for sodium. An equilibrium potential is a theoretical membrane potential. It's what the membrane potential would become if the cell became freely permeable to that ion. Okay, so let's think first about the equilibrium potential for potassium. So the cell is already quite permeable to potassium. And as potassium leaves, you get fewer and fewer cations inside the cell, making the interior of the cell more negative. Right? So if the cell becomes even more permeable to potassium and even more potassium leaves, the cell would become even more negative. Okay? So the equilibrium potential for potassium is even more negative than at rest. Okay, so if you open even more channels for potassium, more potassium leaves, fewer cations inside, means more negative charge inside, lowering the membrane potential. Okay, and because cells are already permeable to potassium, resting membrane potential lies really close to the equilibrium potential for potassium. Let's look at the equilibrium potential for sodium. If you open sodium channels, what happens? Which way does sodium move? Into the cell. And does it move slowly or does it really want to get in? It really wants to get in. It wants to get in so badly that you actually make the interior of the cell positively charged. Okay, so if a cell became freely permeable to sodium, it would go all the way up to positive 60 millivolts. Right? So much sodium would rush in, the interior of the cell would become positive. Right. Everyone fine with this? Because if you understand this, again, action potentials are easy. Okay, we're all set with the resting membrane potential? You guys ready to mess it up? Okay. So let's talk about how neurons change the membrane potential. Okay. Neurons can change their membrane potential because they have gated ion channels. These are ion channels that are not always open. All cells have leak ion channels for potassium. Right? Neurons have gated ion channels that open or close in response to a stimuli. And when they open, ions are going to move down their electrochemical gradients. And because ions are charged, that's an electrical signal. You have the movement of charge. Okay, and it happens rapidly. Okay, so neurons have two major types of gated channels. They've got ligand gated and they have voltage gated. Okay, so the two major types of ion channels that are gated on neurons are ligand gated and voltage gated. A ligand is anything that binds to a receptor. Okay, and ligand gated, usually that ligand is a neurotransmitter. Okay, so a neurotransmitter binds the ligand gated ion channel opens, ions are going to move down their electrochemical gradients and cause a small change in membrane potential. Okay, ligand gated ion channels cause small changes in membrane potential. They cause graded potentials. And we'll talk about graded versus action potentials in just a minute. You can add to this that they cause graded potentials. Right? And a form of graded potentials are synaptic potentials. So these are the changes in membrane potential at a synapse. Okay, so you see lots of ligand gated ion channels on postsynaptic neurons where there's synapses. So that means you see lots of them on dendrites and cell bodies. Okay, so ligand gated ion channels are in high abundance on dendrites and cell bodies. And their ligands are neurotransmitters. Okay, voltage-gated channels open or close in response to a change in membrane potential. And that change in membrane potential is due to those ligand-gated channels. Okay, so the ligand-gated channels cause small changes in membrane potential that then affect the voltage-gated channels. Right? And your neurons have three types of voltage-gated channels: sodium, potassium, and calcium. Sodium and potassium voltage-gated channels are found in high abundance starting at the axon hillock and then all the way down the axon. They're what cause action potentials. Okay? Voltage-gated sodium and potassium channels cause action potentials. And then at the axon terminals, you have voltage-gated calcium channels. And they're what cause exocytosis of neurotransmitter. Or here, I just said release. But neurotransmitters are released by exocytosis. Okay, so that means our neurons, dendrites and cell body, we've got ligand-gated ion channels. Axon hillock, all the way down the axon, we've got sodium-potassium channels that are voltage-gated. And then axon terminal, we have voltage-gated calcium channels. Okay, so different parts of the neuron express different types of ion channels. Okay, we're going to do a little more vocabulary so that we can talk about action potentials. So polarization, a membrane is polarized whenever the membrane potential is other than 0, when there's a difference in charge. Okay, and all living cells are polarized. Okay, if membrane potential is at 0, you've got a dead cell. It's not able to maintain a membrane potential. Okay, depolarization is whenever the membrane becomes less polarized than at rest. And what's resting membrane potential? Minus 70. Okay, so whenever it comes closer to 0 or above 0, that's called depolarization. Repolarization is whenever the membrane returns to rest. Okay, hyperpolarization is whenever the membrane goes below resting membrane potential, becomes more negative than minus 70. Right, and let's look at this in a figure. Okay, so here we've got membrane potential on the y-axis. We have the equilibrium potential for potassium. Right, we've got resting membrane potential at minus 70, and we have the equilibrium potential for sodium. So this would be minus 94 down here, and this would be positive 60. Okay, so we've got a cell at rest, blue dotted line. If it goes above rest, it says it's depolarizing. If it returns to rest, it's repolarizing. And if it goes below resting membrane potential, it's hyperpolarizing. Okay, so what could a cell become permeable to in order to depolarize? What kind of ion channel could open to depolarize the cell? A sodium channel. Yep. What other type of channel? Calcium. Okay, sodium or calcium channels will cause depolarization. The opening of sodium or calcium channels cause depolarization. Okay, how about hyperpolarization? What kind of ion channel would have to open to cause hyperpolarization? Potassium, definitely, or chloride. Okay, so potassium and chloride channel opening causes hyperpolarization. Right? And if you understood that question and came up with the answer, action potentials will be easy. Yeah?
So when it returns to resting membrane potential after hyperpolarization, is there a term for that?
So some textbooks call it repolarization. Some say it's only when it's been depolarized can you call it repolarization. I'm not going to, be nitpicky about it. When it returns to rest, you can call it repolarization. Yep. Just that depolarization is going above rest, hyperpolarization is going below rest. Yeah.
So opening the sodium causes depolarization.
Opening sodium channels would cause depolarization, right? Because sodium wants to enter the cell and it's positively charged, right? It would make the cell less negative. Okay, so here we have another figure showing depolarization, hyperpolarization. So here we have resting membrane potential at -70. This cell has depolarized, it's returning to rest, so it's repolarized, and here it is hyperpolarizing. It's going below rest. Okay, and it's just showing it on a smaller scale. Right, so let's talk about what neurons do. Neurons change membrane potential in two ways. Right? They change it in graded potentials, and they change their resting membrane potential in action potentials. Okay, graded potentials are being shown here. So this would be resting membrane potential right here. You could add -70 right here, where this green line is flat. That'd be resting membrane potential. Here we've got small changes in membrane potential that have occurred. These are called graded potential. They're small changes, and they communicate only short distances. And they occur due to the ligand-gated ion channels. So these are the changes in membrane potential that occur on dendrites and cell bodies. Okay? They're the graded potentials, just small changes. When graded potentials sum, right, and bring the axon hillock to threshold. This is what this -55 is. This is the threshold for the voltage-gated sodium channels that you start to find on the axon hillock. Okay, so when graded potentials sum and bring the axon hillock to threshold, you get an action potential, which is a large change in membrane potential, and it can travel long distances. And it's due to the voltage-gated sodium and potassium channels. Okay, so graded potentials are due to the ligand-gated ion channels. Action potentials are due to the voltage-gated. Graded potentials are fired on the dendrites and cell body. Action potentials on the axon hillock, and then all the way down the axon. Okay, so let's talk about graded potentials first. Okay, so graded potentials get initiated by some sort of stimulus. What we're going to talk about is that stimulus for now being the opening of a ligand-gated ion channel. When we talk about the sensory systems, we'll talk about other type of stimuli that can cause graded potential. But for now, we'll say neurotransmitter binds to ligand-gated ion channel. It opens for a little bit. Ion can move down its electrochemical gradient. Right, but then that ligand-gated ion channel closes. Right, we get a small change. However, the change can vary in magnitude. Okay, graded potentials can vary in magnitude. Action potentials don't. That's why it's an important point. Graded potentials vary in magnitude. The stronger the stimulus, the bigger the graded potential. Okay, so stronger stimulus leads to larger graded potential. And is this showing depolarization or hyperpolarization? Depolarization. Good. Okay. Graded potentials can be depolarizing or hyperpolarizing. If they're depolarizing, so here we have stimulus Z causing depolarization, they're what's called excitatory. They're going to make that neuron more likely to fire an action potential. If they're hyperpolarizing, they're inhibitory. They're going to make that neuron less likely to fire an action potential. And you may be thinking, why the heck would you ever want hyperpolarizing graded potentials? Don't you want your neurons always to fire action potentials? No, you're shaking your head no. Why don't you want your neurons always to fire action potentials?
Like if you receive the pain stimulus,
Uh-huh.
you wanted it like to continually.
True. Exactly. You don't want to continuously feel pain, right? You want to inhibit that sensation. Okay? And the way your neurons inhibit a sensation is through inhibitory graded potentials. So it's important so that you don't feel pain all the time. So that's what your body's endogenous analgesic system is doing. It's trying to inhibit pain signals, but also just regular sensory information. Do you want to pay attention to everything all the time? No, right? Otherwise your socks would itch continuously. Although now everyone's socks are going to itch, okay? You would not be able to sleep, right? Okay? When you sleep, you have these inhibitory inner neurons that are basically stopping signals from going up to your consciousness so that you aren't consciously aware of what's coming in through your ears. Okay, your hearing doesn't shut off. It just doesn't go up to your consciousness. Okay, what do you do to stop light perception? You close your eyes. Yep. Okay. Okay, same thing for descending pathways though too. Okay, you have these inhibitory centers in your brain, the basal nuclei. They get damaged in Parkinson's disease. What's a hallmark of Parkinson's disease? What do people do? They have tremors. They don't want to have tremors, right? They're not telling their hand to do that. It's that their inhibitory inner neurons and their basal nuclei aren't functioning properly. So basically your brain is always sending way too many signals to your muscles. Okay, and those inhibitory inner neurons are preventing all those signals from going so that you have nice smooth movement. Okay, so inhibitory information is very important. Okay, graded potentials are what determine whether or not that neuron is going to fire an action potential. Okay, so graded potentials are occurring on dendrites in the cell body, and then they have to sum up at the axon hillock and either bring it to threshold or don't bring it to threshold. So here we have minus 55, which again is that threshold potential for voltage-gated sodium channels. If you don't bring the axon hillock to threshold, no action potential gets fired. Okay, so these two graded potentials, W and X, weren't strong enough, and Y was actually inhibitory, so no signal going forward. Okay, here we have signal W, right, and it wasn't strong enough to bring axon hillock to threshold, but if it happens over and over, we get what's called summation, brings the axon hillock to threshold, fire an action potential. Signal will proceed. Okay, so graded potentials determine whether or not an action potential will be fired. So they're very important. Right, graded potentials spread by what's called electrotonic conduction, which is just passive charge movement. Okay, so here we've got the stimulus. Say it's a ligand-gated sodium channel. Okay, so a neurotransmitter binds, a ligand-gated sodium channel opens, sodium rushes in, and then it's just going to diffuse away. So the thicker the arrow, the higher the concentration of sodium. Okay, so the change in membrane potential at the site of the stimulus, here's the graded potential. Here is the change in membrane potential at the axon hillock. Notice the change in membrane potential is less, right? Because as the charge spreads, it decreases. Okay, so graded potentials are decremental. Magnitude decays as it spreads. Okay, this is an important point to think about though, because synapses, the closer the synapse is to the axon hillock, the stronger that synapse is, right? Because the charges don't have to move as far. Okay, so the closer the synapse is to the axon hillock, the stronger it is. One of the things you do when you learn and form memories is that you make new synapses. And the more you practice information, the stronger those synapses will be. Okay? So when you study, you're actually rearranging synapses in your brain. As long as you just don't cram right before the exam. When you cram, you don't do that synapse formation. Okay, you got to study multiple days in advance, and you've got to repeat the information over and over again. Okay, and young brains do this faster and easier than old brains. And this is why we send you to college when you're still young. Okay, not that you can't come to college when you're older. You just have to study harder, and it stinks. Okay, older brains don't learn as fast as younger brains. Okay, graded potentials, I already mentioned this, can sum. Okay, action potentials don't sum. Graded potentials can sum, and they sum in one of two ways. Temporally, so that's the same stimulus happening really fast over and over, or spatially, this is where you get different stimuli that overlap in time. So up here, we have temporal stimulation. We already looked at this figure. Okay, so this is the same ligand-gated ion channel opening over and over again. Okay, so when it stays open for a longer period of time, let's say even more sodium can get in, we bring the axon hillock to threshold, action potential fires. Okay, spatial summation, we have a synapse for W and a synapse for X. When they both open at the same time, enough cations come in that it brings the axon hillock to threshold. Okay, so this would be spatial summation. Spatial summation, you can also have a depolarizing or excitatory graded potential, a hyperpolarizing and inhibitory graded potential occur at the exact same time, right? And it basically flatlines. This cell is not going to come to threshold. Okay? So they can cancel each other out, or they can increase the signal. So, graded potentials cause action potentials. Action potentials are brief changes in membrane potential. They happen really fast, and they're large. They're 100 millivolt changes. Okay, graded potentials are small. Okay, they're anywhere from maybe 5 millivolts up to 25 millivolt changes. Okay, during an action potential, membrane potential actually reverses. The interior of the cell becomes positive. Action potentials, unlike graded potentials, do not decrease in strength as they travel. Okay, so they do not decay. So they can travel long distances. They do not sum, and they're always excitatory. So they differ from graded potentials in those ways. Right? We will watch the
Neurons send signals over long distances by generating and propagating action potentials. Most action potentials originate near the axon hillock of the cell body, in the initial segment of the axon. It then travels the entire length of the axon. A closer look reveals that during an action potential, voltage-gated channels open and close, altering the permeability of the plasma membrane to sodium and potassium ions. A threshold stimulus changes the shape of the voltage-gated sodium channels, causing their activation gates to open. This event marks the beginning of phase 1 of the action potential, known as depolarization. As sodium ions diffuse into the axon, the membrane potential becomes less negative. This causes more voltage-gated sodium channels to open, and the membrane potential soars to plus 30 millivolts. At this point, two key events occur. The inactivation gates of voltage-gated sodium channels close, and voltage-gated potassium channels open. These two events mark the beginning of phase 2 of the action potential, known as repolarization. As potassium ions diffuse out of the axon, the membrane potential becomes negative again. However, the membrane potential continues in the negative direction, going beyond the resting state of minus 70 millivolts. This marks the beginning of phase 3 of the action potential, known as hyperpolarization. During this phase, voltage-gated potassium channels close and all voltage-gated sodium channels are released from inactivation. By the end of this phase, ions move through leak channels only and the membrane potential returns to the resting state of minus 70 millivolts. The neuron is now ready to fire another action potential. Summary: generation of an action potential. A threshold stimulus opens voltage-gated sodium channels. Sodium ions diffuse into the axon, depolarizing it to plus 30 millivolts. Voltage-gated sodium channels close, and voltage-gated potassium channels open. Potassium ions diffuse out of the axon, repolarizing it to a negative value. The membrane potential briefly hyperpolarizes. Voltage-gated potassium channels close, and the membrane returns to the resting state of minus 70 millivolts.
So we just watched that video talking about action potentials, and now we'll go through step by step. Right, so here we have membrane potential on the y-axis and time in milliseconds on the x-axis. Okay, so action potentials are really fast. They occur within milliseconds. And we're going to separate the action potential into 3 phases. Phase 1, which is depolarization. Okay, phase 2, which is repolarization, and phase 3, which is after hyperpolarization. Okay, now we have two dotted lines on this figure. One's at minus 70 millivolts, that's resting membrane potential, so V subscript M. And then we have minus 55 millivolts, which is the threshold potential for the voltage-gated sodium channels. Okay, so at minus 55 millivolts, they open, and that's what starts phase 1, or the depolarization phase, okay, which is due to sodium influx. Right, when those sodium channels open, which way does sodium want to move? Into the cell, down its electrochemical gradient, and it rushes in. And it rushes in so fast, it actually makes the interior of the cell positive, right? It peaks at positive 30 millivolts. So much sodium rushes into the cell, the interior of the cell becomes positive. Okay? At the peak, those inactivation gates, which are mentioned in the video, shut. And once they shut, sodium can't rush in. And what opens at peak are voltage-gated potassium channels. And those are the channels that cause potassium efflux. So all of a sudden, the cell becomes highly permeable to potassium. Right? And which way does potassium move? Out. Right? The interior of the cell at this point is positive. So this means potassium's electrical and chemical gradient is to leave the cell. So it rushes out, right? It doesn't slowly leak out, it rushes out. Okay? Those potassium channels stay open after the membrane hits resting membrane potential. So at this point, the cell is even more permeable to potassium than at rest. So even more potassium leaves, and that's what causes the hyperpolarization. Finally, those potassium channels close. The sodium-potassium pumps have been working this entire time. They re-equilibrate everything back to rest. Okay, so our three phases are depolarization, repolarization, after hyperpolarization. Yes?
Can you go over the hyperpolarization again? You said something about
Yes.
potassium channels, something.
Exactly. Okay. So at the peak of the action potential, voltage-gated potassium channels open. Okay, so their threshold is positive 30. The threshold for the voltage-gated sodium is minus 55. They open, and then their inactivation gate closes, voltage-gated potassium open, and they stay open for a couple milliseconds, right? In the meantime, potassium rushes out, and because you now have both leak potassium channels open and voltage-gated potassium channels open, the cell hyperpolarizes. Because where's the equilibrium potential for potassium? Who remembers? Negative 90 or negative 94, depending on what figure you look at, right? Which is below minus 70. So basically, the cell is now freely permeable to potassium. So it almost hits the equilibrium potential for potassium. Okay, and that's what causes that after hyperpolarization. So the fact that those voltage-gated potassium channels are still open. Okay, they've hyperpolarized that portion of the membrane. Okay. So what this figure is showing is, again, we have membrane potential. So that same y-axis we were looking at in the previous figure. Okay, so the action potential here is being shown in red. So it looks identical. We've got depolarization, repolarization, after hyperpolarization, back to rest. But we have an additional y-axis over here, which is membrane permeability. And membrane permeability is for potassium, which is the dark blue line, and did I say potassium or sodium? Sodium! You guys, this is the dark blue line is for sodium. Sorry about that. The dark blue line is for sodium, and the teal line is for potassium. So now we're looking at how permeable that portion of the neuron is to sodium versus potassium. So notice at rest, permeability to potassium is greater than sodium. Basically, the neuron's not permeable to sodium, but it is permeable to potassium because of those leak channels. Okay? At minus 55 millivolts, voltage-gated sodium channels open. So look at this. We get this huge increase in permeability of sodium. And that parallels the depolarization phase. Okay, so the cell becomes permeable to sodium. At peak, right, those voltage-gated potassium channels open. So the permeability of the cell to potassium increases as the permeability to sodium plummets. Right, so potassium is rushing out, and notice that permeability to potassium stays past rest. So even more potassium can leave the cell, and that's what causes the hyperpolarization. As soon as those voltage-gated potassium channels close, all you have are the leak channels open, right? And then the sodium-potassium pump is going to re-equilibrate the membrane to rest. Okay, so depolarization is due to sodium influx. Repolarization is due to potassium efflux. Yes?
So you said that at negative 55, the sodium channels open.
Yes.
So how does the cell get from negative 70 to negative 55?
Good question. How does the cell get from minus 70 to minus 55? That's an excellent question. Who knows the answer? Yeah.
They're graded potentials.
Exactly. Graded potentials. Yes. Okay. Graded potentials are what bring the axon hillock to thresholds. Okay. Let's talk a little bit about those sodium channels. The video mentions this, right? The sodium channels, those voltage-gated sodium channels have two gates. Okay, they have a voltage gate and they have an inactivation gate. The voltage gate at minus 55 millivolts opens. Okay, so the voltage gate opens at minus 55 millivolts. When the voltage gate is open, sodium can rush in. And that's going to cause the depolarization phase. But a millisecond after the voltage gate opens, the inactivation gate swings shut. Okay, so those sodium channels only stay open for a millisecond. Right? And as soon as the inactivation gate shuts, no more sodium can enter. Right? So voltage gate opens at minus 55 millivolts. Then a millisecond later, sodium gate slams shut. Inactivation gate slams shut. Okay, and it stays closed until the cell goes back to resting membrane potential. Okay, until it crosses to or below resting membrane potential. Because at that point, that voltage gate will be shut again. Right, so how does the axon hillock get to threshold? Gets there by those graded potentials. So here we've got, remember, graded potentials are small changes in membrane potential, and they vary in magnitude. Right, so we can have a small stimulus causing a small graded potential. So here's a subthreshold stimuli causing too small of a graded potential. Another, it's slightly stronger, causes a slightly larger graded potential, but again, not strong enough to bring the axon hillock to minus 55. Here we finally have a threshold stimulus, okay, that has brought the axon hillock to minus 55 millivolts. And this could be a single stimulus that's strong enough, or remember, graded potentials can sum. So it could be multiple stimuli, summing spatially, or the same stimuli happening over and over again and summing temporally. Okay, but as long as the stimulus brings the axon hillock to threshold of minus 55 millivolts, it's called a threshold stimulus. Okay, and you fire an action potential. If the stimulus gets even stronger, it's called a suprathreshold stimulus. Okay, but look, you fire the exact same action potential. Action potentials do not vary in magnitude. Okay, so on the next exam, as well as on some quizzes, I'm going to say, something about action potentials, and I often use a distractor where I say you get a larger action potential or a smaller action potential. It's never the correct answer. Right? Action potentials are always the same. They're always 100 millivolt change. They do not vary in magnitude, no matter how strong the stimulus is. Okay? Action potentials do not vary in magnitude. They vary in frequency. Okay, so stronger stimuli will cause higher frequency action potentials than weaker stimuli. And we'll talk about why. Okay, so action potentials are all or none. If the threshold isn't strong enough, no action potential. If the threshold is strong enough, action potential. If the stimulus is super strong, same action potential, right? No variation in magnitude. No matter if you get a threshold or a super threshold stimulus, you always get 100 millivolt change in membrane potential. Okay, that's the all or none principle. You either get an action potential and it's always the same, or you don't get one. Okay, action potentials have what are called refractory periods. They're time periods where you have decreased excitability. And we're going to talk about two refractory periods, the absolute refractory period and the relative refractory period. Absolute, think of as being when there's absolutely no more action potentials. Okay, during the absolute refractory period, more action potentials cannot be fired. During the relative refractory period, think of it as being relatively harder to fire another action potential. Okay, you can fire another action potential during the relative refractory period. You just have to have a suprathreshold stimulus. Okay, so the absolute refractory period spans all of depolarization and most of repolarization. Okay, during that time, second action potential can't be fired, and this is all because of the sodium channels. It's either because they're already open, they can't open anymore, or that inactivation gate is shut. Yes?
In reference to this, what happens when you have a constant stimulus, such as like sight or music?
Oh, it's all about frequency. We'll talk about that. You can have different frequency of action potentials. So you can fire so many action potentials per second, right? And the stronger the stimulus, the more action potentials per second you'll fire.
So does that mean that we actually hear like you know, like almost dots because it's going up and down?
No. Hearing is super complicated.
Okay.
We're going to talk about it a little bit when we talk about sensory systems. But yeah, so basically all your sensory information is being coded into action potential frequencies that then your brain decodes as sensory information. Yeah. It's sort of like computers are all ones and zeros, right?
Oh.
Yeah. Uh-huh.
Is there a limit on frequencies?
Is there a limit? Yes, the absolute refractory period puts the limit on it, right? Because during the absolute refractory period, you can't fire another action potential. So you can only fire super high frequency where you get second firing in the relative refractory period, okay? So, relative refractory period is when the cell is hyperpolarized due to those voltage-gated potassium channels being open, right? So if it's hyperpolarized, it takes a stronger stimulus to bring it to threshold. That's why it's relatively harder to fire a second action potential. Look at them in a figure. Right here we've got our friend, the action potential again. Okay, we're just showing a single one at this point. We got membrane potential. This would be minus 70. This would be positive 30. Right? And here the blue lines are showing where is the absolute refractory period versus the relative refractory period. Okay? And in this bottom figure, we have the percent of sodium channels inactivated, okay, versus the percent of voltage-gated potassium channels open. So the red line versus blue line are showing us different things. Percent sodium channels inactivated means they have the inactivation gate closed. When the inactivation gate is closed, it's not going to open again. Sodium cannot come in. You cannot fire a second action potential. So that's leading to the absolute refractory period. Okay? You can't open sodium channels. If you can't open sodium channels, you can't depolarize anymore. Okay? The relative refractory period is due to the fact that you have voltage-gated potassium channels still open, which is hyperpolarizing the cell. Okay, so the cell is even farther from minus 55 millivolts, which would open voltage-gated sodium channels. Okay, so you need a stronger stimulus to fire another action potential. All right. Any questions about action potentials? We're going to talk about them more. We're just going to talk about them propagating down the axon. But questions about how they're fired? Yes?
So we talked about how an action potential is always 100 millivolts.
Yes.
So when it's in that hyperpolarization, it needs a stronger stimulus, like to get to the negative 70 and then to get to the 30.
Yes, okay. So, yeah, if you that's quite the technicality. So it's true. When it's hyperpolarized and it's closer to minus 90, we fire a second action potential, it goes all the way up to positive 30.
Okay, so does it go like 20?
No, it doesn't go 20.
Okay.
Oh, that's what your question was. No. Okay, so it is true. If you fire to the action potential while the cell is hyperpolarized, that net change might be slightly higher than 100 millivolts. That is true. Yes. So we could say action potentials always peak at positive 30 millivolts. Yeah. Okay. Any other questions? Some of you guys are paying really close attention, which is good. All right. Right. So where are action potentials initiated? Axon hillock. Okay. Once they're initiated at the axon hillock, they're going to travel from the axon hillock towards the axon terminals. Okay? Action potentials are unidirectional. Right? They travel in one direction from the axon hillock where they're initiated down to the axon terminals. Okay, so here we have a motor neuron. Okay, so dendrites and cell body would be in the central nervous system, as with the axon hillock. We have a bunch of graded potentials occurring on the dendrites and cell body. If it sums and brings the axon hillock to threshold, action potential fires, travels all the way down the axon. This axon's myelinated, so it's actually going to hop from node to node, basically. Okay, and then at the axon terminal, it's going to cause the release of neurotransmitter. Here we have a sensory neuron. So we've got the dendrites, right? And they have graded potentials occurring on them, which then sum, and here's the axon hillock right here. Okay, and so the axon hillock would be brought to threshold. Action potential would go down the axon towards the axon terminals. Okay, and in sensory neurons, the cell body is not associated with the dendrites. Okay, we're going to talk about two types of propagation. One is called contiguous, and that is not a typo. You can think of it as being continuous, but it's actually called contiguous. And this is how unmyelinated neurons fire action potentials. So if the axon is unmyelinated, every portion of the membrane fires an action potential. So it's slower. Right? Saltatory conduction is what happens in myelinated fibers. Okay, so that myelin acts as insulation, and only the nodes of Ranvier where there's no myelin, those are the only areas of the axon that fire action potentials. And then the electrical current spreads under the myelinated portions really rapidly. Okay, so myelination speeds conduction. Okay. Unmyelinated neurons fire slower action potentials, or it's slower for the action potential to go from the axon hillock to axon terminal, because every portion of the membrane has to fire an action potential. Whereas in myelinated, it jumps from node to node, so much faster conduction. Right, so we'll watch another.
Action potentials propagate in a continuous fashion in unmyelinated axons. Once an action potential is generated in the initial segment of the axon, it propagates the entire length of the axon. Recall that a threshold stimulus causes voltage-gated sodium channels to open. The influx of sodium ions generates an action potential. It also establishes a depolarizing current that flows to the next segment and brings it to threshold. Voltage-gated sodium channels open, regenerating the action potential in this segment of the axon. Current flows from this segment and depolarizes the next segment to threshold, thus regenerating the action potential yet again. In this way, regeneration continues in one direction all the way down to the axon terminals. The basis for unidirectional propagation is revealed when we take a closer look. By the end of the depolarization phase of the action potential, all voltage-gated sodium channels inactivate and voltage-gated potassium channels open. These two events render this segment of the axon temporarily insensitive or refractory to another depolarizing stimulus. However, voltage-gated sodium channels in the downstream segment are closed and receptive to a depolarizing stimulus. Thus, propagation occurs sequentially down the axon to the axon terminals. In myelinated axons, action potential propagation is a bit different. Here they propagate in a saltatory or leaping fashion. The myelin sheath consists of multiple layers of tightly wrapped glial cell membrane. But this sheath is not a continuous one. Exposed areas of axonal membrane, known as nodes of Ranvier, occur at discrete intervals. Voltage-gated sodium channels are abundant in the nodes, but largely absent between nodes. So, action potentials are regenerated at each node, not in areas covered by the myelin sheath. However, the myelin sheath does provide the insulation necessary for the rapid spread of depolarizing current. And the sooner the nodes reach threshold, the faster action potentials propagate along the axon. Saltatory conduction is extremely fast. Velocities often exceed 100 meters per second. In contrast, continuous conduction is fairly slow. Velocities rarely exceed 2 meters per second. Nevertheless, both continuous and saltatory conduction propagate action potentials over varying distances because action potentials regenerate along the way. Summary: propagation of an action potential. Once generated, the action potential propagates the entire length of the axon without decrement.
All right. So the video did a really nice job talking about contiguous versus saltatory. And let's look at contiguous first. So talked about the fact that you get unidirectional flow due to the absolute refractory period. So here we have the axon hillock, right? This is part of the soma up here. And this neuron is not at threshold, okay? It's at rest. So this is panel A. Here's panel B. Here, graded potentials occurring in the soma or the dendrites have brought the axon hillock to threshold, right? Voltage-gated sodium channels have opened. Sodium has rushed in. The interior of the cell is now positively charged. Okay, those positive cations are going to diffuse away. Some of them are going to diffuse up into the soma, and some of them are going to diffuse towards site B or downstream. Okay? Why doesn't the soma fire an action potential?
It's in refractory.
Is it in refractory? Did it ever fire an action potential? It can't fire an action potential. Why can't it fire an action potential? What doesn't it have? Voltage-gated sodium and potassium channels. What kind of channels does the soma and the dendrites have? Ligand-gated, right? So the reason why the soma and dendrites don't fire action potentials is because they can't. They don't have those voltage-gated sodium and potassium channels. Those are found on the axon hillock and all the way down the axon. Okay, so even though a ton of sodium rushes up into the soma, it doesn't have voltage-gated sodium channels. It's not going to fire an action potential. Okay, site B is going to. So let's go up here to panel C. Okay, in panel C, site B is now firing action potential. Its voltage-gated sodium channels are open. Sodium rushes in. Some of it moves upstream to site A. Some of it moves downstream to site C. Now, why does site A not fire an action potential? It's in a refractory period. Okay, those inactivation gates are shut on the sodium channels. It can't fire an action potential. Site C, however, can. So it fires an action potential. Okay, some of the sodium comes down to site D, some to site B. Okay, site B is in refractory, so only site D fires an action potential. And it continues like that on and on and on and on, all the way down the axon. Okay, that's contiguous conduction. Okay. Myelinated fibers have action potentials that travel 50 times faster than an unmyelinated neuron of the same size. Okay, so significant increase in conduction velocity. And that's because only the nodes of RANVA fire the action potentials. Right? Myelin acts as insulation because it's primarily lipids, and charged substances can't cross lipid bilayers. Okay, so myelin is made by two different types of glial cells. Oligodendrocytes are glial cells found in the central nervous system, and they myelinate interneurons. Okay, so oligodendrocytes myelinate interneurons. And one oligodendrocyte will myelinate multiple interneurons. In the peripheral nervous system, Schwann cells myelinate those peripheral neurons, both efferent and afferent neurons. Okay, and a single Schwann cell completely wraps itself around one neuron. Here we can look at that. Here we have oligodendrocytes versus Schwann cells. Okay, they're both wrapping their membranes around neurons. It's just that oligodendrocytes will wrap multiple neurons. So here, this single oligodendrocyte is wrapped around 3 different axons. This allows for some amount of crosstalk within the central nervous system. Okay, in the peripheral nervous system, this would be a motor neuron, so it would be an efferent neuron. The Schwann cells completely wrap themselves around a single axon. Right? Both leave spaces, and those spaces are called nodes of RANVA, and that's where the action potentials get propagated. Okay? Underneath, you get really fast current spread because this acts as insulation. Right? So the sodium that rushes in at this node is going to spread really quickly underneath the myelin sheath and bring the downstream node of RANVA to threshold. Right, so here we've got a motor neuron. Here's the axon hillock. Okay, and then we have Schwann cells myelinating it. And so here we have one of the nodes firing an action potential, right? And that sodium that rushes in is going to spread and then bring the downstream node to threshold. I don't really like how this figure makes it look like the sodium only goes in one direction. That's not true. Okay, it would also spread upstream, but this node would be in a refractory period. Okay, so only the downstream node would be available to fire another action potential. Okay, and then under the myelin, you're going to have really fast current spread. Okay, so much faster, 50 times faster. Yeah?
Does any of this current mesh with the membrane potential of the myelinated sheath?
So, oh, you mean does the actual Schwann cell, does its membrane potential change?
Yeah.
No. No, because there are no channels in the area that's myelinated, and there's no sodium-potassium pumps. Right? So basically, all those ions that rush in get trapped underneath the myelin. Okay? So they're going to rush towards the nodes because that's where they can get kicked out. Right? Right. Any questions about propagation? Okay, let's start in and talking about the synapse then. Okay, so we're going to talk about two different kinds of synapses, although the most common kind are chemical. Right? A synapse is an association with the neuron and another neuron or with an effector organ. And that would always be a muscle, could be cardiac muscle, smooth muscle, or skeletal muscle, or it's going to be a gland. Right? And there's two types of synapses. There are electrical synapses and there are chemical synapses. But chemical synapses are the most common type of synapse. Right? With electrical synapses, you actually have direct connection between a neuron and another neuron, or a neuron and an effector organ, via gap junctions. Okay, so pores between the cells. And that allows ions to spread directly from one cell to another. So no neurotransmitter is involved in electrical synapses. No neurotransmitter is involved in electrical synapses. Right? Electrical synapses are really rapid, super fast. They also allow for bidirectional communication. So this means the presynaptic cell can excite the postsynaptic cell, and the postsynaptic cell can excite the presynaptic cell at the exact same synapse. Which is bad if you want to develop circuits. Okay, so if you want really complex circuits in order to control complex behaviors and physiological functions, you don't want bidirectional communication. Okay, so this is why we see chemical synapses more commonly, right? Because we want the presynaptic cell to talk to the postsynaptic cell in the same way all the time so that we can develop really complex circuits. Okay, the same synapse can also be excitatory or inhibitory. Right? We see some of these electrical synapses between neurons and their glial cells. So like between neurons and oligodendrocytes, you might see some electrical synapses, or neurons and astrocytes. Right? But the vast majority of synapses between neurons and other neurons and effector organs are chemical. Right, so we're going to talk about chemical synapses today, and these are the most common types of synapses, and you have chemical synapses between two neurons. You have chemical synapses between neurons and muscles, and those muscles could be skeletal, smooth, or cardiac, right? And then you can have chemical synapses between neurons and glands. And with a chemical synapse, the action potential doesn't spread directly from cell to cell like in an electrical synapse. Right? You've got release of neurotransmitter from the presynaptic neuron, and then that neurotransmitter has to bind to a receptor on the postsynaptic cell. Okay, so you've got a release of a chemical messenger, and that chemical messenger is a neurotransmitter. Okay, so here we've got some examples of neuron to neuron synapses. Okay, so here we've got presynaptic neurons. This presynaptic neuron is synapsing on the postsynaptic neuron's dendrite. So this is called an axodendritic synapse. So the axon of the presynaptic neuron is synapsing on the dendrite of the postsynaptic neuron. Here we have a presynaptic neuron that is synapsing on the soma. So this is an axosomatic synapse. Okay, so the axon of the presynaptic neuron is synapsing on the soma of the postsynaptic neuron. Okay, question for you guys. Which one of these synapses on this figure, okay, this is not saying all the time, but on this figure, is the axodendritic synapse or the axosomatic synapse stronger? Which one is a stronger synapse? Axon somatic. And why do you say that? Because it's closer to the axon hillock. Okay? I'm not saying that all axosomatic synapses are stronger, because you could have had an axodendritic synapse that synapsed on the dendrite right here, right? And that's close to the axon hillock. Right? But in this figure, this synapse is stronger because it's closer to the axon hillock, because the graded potential that will result at the synapse decays as it travels. So you want synapses, if you want them to be strong, you want them as close to the axon hillock as possible. Right? And you make new synapses. Right? When you learn and form memories, it involves new synapse formation. And when you practice material, right, it'll involve making stronger synapses so that you can recall it more quickly. Here's a third kind of synapse. So here we have a presynaptic neuron, and it's synapsing on the axon of the postsynaptic neuron. So this is an axonic synapse. And this is often what inhibitory neurons do. Right? So they're going to inhibit the signal right at the axon terminal. Okay, so inhibitory neurons often do this, prevent that neuron from sending the signal. So we have inhibitory interneurons that are really important for sleep and our reticular formation, right? And they can stop sensory information from being propagated. Right, by stopping that action potential before you get the release of neurotransmitter. All right, so let's look at the anatomy of a synapse. Okay, so we have a presynaptic neuron here and the postsynaptic neuron. The presynaptic neuron, we're showing a blowup of the axon terminal. In the axon terminals, we find voltage-gated calcium channels. Okay, what kind of channels are on the rest of the axon? Voltage-gated sodium and potassium channels. Okay, on the axon terminal, instead we get voltage-gated calcium channels. Right, when an action potential reaches the axon terminal, it opens those voltage-gated calcium channels. And where's calcium in higher concentration? Outside of the cell. And so what does calcium do when a calcium channel opens? Rushes in, okay, and that intracellular calcium acts as a signaling message. I don't know what I'm trying to say. It acts as the signal, right, to cause the synaptic vesicles that are filled with neurotransmitter. Okay, so these little triangles are neurotransmitter. It tells those synaptic vesicles to move to the plasma membrane and release the neurotransmitters by exocytosis. Right? So these synaptic vesicles are filled with neurotransmitter. All neurotransmitters are water soluble so that they can be held in vesicles. Okay? And they're released by exocytosis. Then we have what's called the synaptic cleft. And that's the space between the presynaptic neuron and the postsynaptic neuron. So no direct connection. There's a space. That space is filled with extracellular fluid. So if we're talking about synaptic clefts in the central nervous system, that space is filled with cerebral spinal fluid. And if we're talking about synaptic clefts in the peripheral nervous system, it's filled with interstitial fluid. Okay, on the postsynaptic cell, we have to have a receptor for the neurotransmitter. Otherwise, there's going to be no response. Okay? Sometimes, okay, so there's always a receptor. Sometimes there's an enzyme that actually breaks down the neurotransmitter. Not all the time, but sometimes. Okay? And then sometimes on the presynaptic cell, there's what's called a reuptake molecule in order to reabsorb the neurotransmitter. Sometimes, but not always. Right, so here we have a step photo showing the steps of communication at the synapse. Okay, so step number 1 is action potential reaches the axon terminal. And we talked about propagation of action potentials on Wednesday. When the action potential reaches the axon terminal, that triggers the opening of voltage-gated calcium channels. So step 2, voltage-gated calcium channels open, and there's many, not just one. Okay, there's many voltage-gated calcium channels. Calcium rushes in down its electrochemical gradient, and that acts as the signal to cause exocytosis of neurotransmitter. So these synaptic vesicles fuse with the plasma membrane and do exocytosis of the neurotransmitter. Step 3. Okay, now we have neurotransmitter in the synaptic cleft. Because neurotransmitters are all water soluble, they just diffuse across the synaptic cleft. Okay, so that's step 4. Neurotransmitter diffuses, and then it binds to the receptor on the postsynaptic cell. Step number 5 is really vague. We get some sort of response in the cell. Okay, there's a response, right? Step number 6 also differs. Okay, step number 6 is the neurotransmitter has to be removed from the synapse, and it's removed either by degradation of an enzyme on the postsynaptic cell. Okay, so our muscles, our skeletal muscles, have an enzyme on their plasma membrane called acetylcholinesterase, which breaks down the acetylcholine that motor neurons release. Okay, so that would be an example of degradation. Okay, or you could have reuptake. So some of our interneurons in our brain use the neurotransmitter serotonin, and the neurons that release serotonin have a reuptake molecule called serotonin reuptake molecule. Okay, and one of the most common classes of antidepressant drugs are SSRIs. You probably heard that before. That's selective serotonin reuptake inhibitor. Okay, so those SSRIs block the reuptake molecule for serotonin, and that leaves serotonin in the synapse for a longer period of time. So you get a more prolonged response in the postsynaptic cell, and hopefully that prolonged response is a sense of happiness and well-being, right? But when you use SSRIs and you leave serotonin in the synapse, you just put on more receptors for serotonin as well. So that's why you're not supposed to go off of SSRIs really quickly. Right? You're supposed to decrease your dosage over a long period of time so that you'll downregulate the number of receptors. Okay? Because if you just go cold turkey on SSRIs, you're going to have all these receptors for serotonin, but you're not going to block the reuptake anymore, and so you're only going to have serotonin in the synapse for a short period of time. You're going to reabsorb it really quickly, and you're going to get no response from the postsynaptic cell. Right? So that's why if you go cold turkey on those SSRIs, often causes, major depression, suicidal thoughts, etc. Okay, got to slowly go off of them. Okay, diffusion is also a possibility, right? You've got a water-soluble molecule in extracellular fluid. It could just diffuse away, right? And one of the major roles of astrocytes, remember those are some glial cells that are in the central nervous system, is to absorb any neurotransmitter that is diffused away from synapses so that it doesn't go and hit another synapse, right? So that's one of the major roles of astrocytes. Can be excitatory or inhibitory. Excitatory synapses cause the postsynaptic neuron to have depolarizing graded potentials. Right? Inhibitory synapses cause the postsynaptic neuron to have hyperpolarizing graded potentials. Okay? Depolarizing graded potentials are going to make that postsynaptic cell more likely to fire an action potential because it's going to bring that postsynaptic cell closer to threshold. Inhibitory synapses, those hyperpolarizing graded potentials, are going to bring that postsynaptic neuron farther from threshold, so make it less likely to fire an action potential. So let's look at both types.
Our brains are constantly processing information, which requires communication between billions of nerve cells. Signals are passed from a sending neuron to a receiving neuron at a junction called a synapse. An action potential in the sending neuron travels down the axon until it reaches a synaptic terminal. The narrow gap between the synaptic terminal and the receiving neuron is called the synaptic cleft. The synaptic terminal of a sending neuron contains numerous vesicles filled with neurotransmitters, chemicals that carry information across the synaptic cleft. When an action potential reaches the synaptic terminal, the vesicles fuse with the plasma membrane of the sending neuron, releasing neurotransmitters into the synaptic cleft. The neurotransmitters affect the receiving neuron, changing the distribution of charge across its membrane. Let's take a closer look. An action potential is propagated down an axon by the opening and closing of sodium and potassium channels. When an action potential arrives at the synaptic terminal, it causes the opening of calcium channels, shown in green. Calcium ions enter the synaptic terminal through the calcium channels. Calcium ions bind to the vesicles containing neurotransmitters. This causes the vesicles to fuse with the plasma membrane of the sending neuron, releasing neurotransmitters into the synaptic cleft. The neurotransmitters diffuse across the synaptic cleft and bind to receptors in the plasma membrane of the receiving neuron. Here, the receptors are ion channels that open. Ions move across the membrane, changing the distribution of charge across the membrane. The neurotransmitters are quickly removed from the synaptic cleft, ending their effect on the receiving neuron. A single neuron can receive signals from many sending neurons. The signals arriving from the axon on the left are excitatory. They make the receiving neuron more likely to generate an action potential, as indicated by the green glows. Signals from the axon on the right are inhibitory. They make the receiving neuron less likely to generate an action potential, as indicated by the red glows. The two sets of signals cancel each other out, and no action potential is generated. Now, only excitatory signals are transmitted, and the receiving neuron generates an action potential down its axon. The signals that pass between these and countless other neurons constitute our thoughts and coordinate our activities.
Okay, so excitatory. Oh yeah, question.
I'm just kind of wanting to know a little bit more. What causes inhibitory signals? I mean, I get excitatory, but what causes? I can't figure out what that would be.
You mean why are there inhibitory signals?
Right. Like
Yes.
excitatory, I want to lift something up, but why?
Right.
Do you have?
Right, why do we have inhibitory signals? So I mentioned earlier we have these inhibitory interneurons in the reticular formation of our brain stem, and that plays a really important role in sleep. So we want to inhibit sensory information from going to our consciousness so that we can sleep, right? So for sleep, super important. Also, so that you can pay attention. Okay, right now you guys are getting sensory information from all of your senses because no one is sleeping at the moment. Okay, right? But hopefully you're paying attention to auditory and visual information and not paying as much attention to your somatosensory information. Okay, so you're not feeling itchy, right? Or your pants don't feel too tight. Okay, and instead you're focusing in on specific sensory information. So inhibiting some sensory information while allowing others through is also super important. Okay. Also descending. So in Parkinson's disease, right, you have destruction of these inhibitory centers in the brain, right, and those inhibitory interneurons aren't working as well, so you get the tremors. So you get unwanted muscle movement, right? So your brain is basically way overactive, and you need to dampen it down.
Could it also be like a process of slowly letting something down? Like if you lift something up?
Yes, so slow methodical movement involves both excitatory information and inhibitory information. So that inhibitory information is occurring in the central nervous system. Okay. So that only specific motor neurons are firing, and firing at a specific rate. Okay. Right? Yeah. Okay. So inhibitory information is often within the central nervous system. Yes?
So is like inhibitory, like when you first get a hot tub, it's really hot, and then like while you're in there, it's cold, like it feels more normal.
Right, so your thermoreceptors are no longer sending, it's either your thermoreceptors are, oh, now we're getting into sensory information stuff. So there's different types of sensory receptors. Some of them are tonic and always send a signal, and some of them are phasic and stop sending a signal. Okay, and if it's always tonic, then you have to have inhibitory interneurons within the central nervous system to turn off that signal. If it's phasic, that sensory receptor just stops sending a signal. So a lot of your thermoreceptors are actually phasic. Same thing with your sense of smell. That's why you walk into a room and you think it kind of stinks, right? But after 5 minutes, you're like, oh, I guess it doesn't smell so bad, right? It's not because the smell's gone away. It's just that your nose has stopped sending a signal to your brain. Other question?
Yeah, I was just wondering where the neurotransmitters come from. Like, do they travel down the axon?
Excellent question. So the neurotransmitters have to travel down the axon because they're made in the soma. Right? So a lot of neurotransmitters are small peptides or amino acids, and those are coming from the soma. So they're getting packaged in the Golgi complex into their secretory vesicles, right? And they're being sent down the microtubules all the way down to the axon terminal. Okay? In neurons that have reuptake molecules, right? Those are getting repackaged in the axon terminal. Okay, but in neurons that don't have reuptake molecules, you're going to have the secretory vesicles coming all the way down the axon. Okay, any other general neuron questions? Yes?
So with any sort of movement, you have both excitatory and inhibitory kind of firing at the same time then?
In your central nervous system. Yep, but our skeletal muscles work by excitatory only. So there's either a signal and they contract, or there's no signal and they don't contract. Okay? Yep, so all that inhibitory stuff is going on in the central nervous system. Okay. Right, so excitatory synapses, right, are the ones that bring the postsynaptic cell closer to threshold. And so an excitatory synapse, the neurotransmitter is going to cause the opening, right, of what are called non-specific cation channels. And a non-specific cation channel lets both sodium and potassium through. Right, and remember, you've got sodium wanting to come in the cell down its chemical and electrical gradient. Right? Whereas potassium wants to leave down its chemical gradient, but it wants to stay in due to its electrical gradient. Okay, so if you open a nonspecific cation channel, way more sodium rushes in than potassium leaves, because potassium has opposing electrical and chemical gradients. Okay, so even though you're letting sodium out of the cell, these type of ion channels are still depolarizing because sodium's electrochemical gradient is so strong into the cell. Right? So non-specific cation channels depolarize the postsynaptic cell. Okay? Generally, one excitatory synapse, activating it, isn't enough. You have to have summation. Right? Either temporal or spatial summation. Okay. The graded potential, that results from an excitatory synapse gets a special name, and it's an excitatory postsynaptic potential, or an EPSP. Okay, just a graded potential that results at an excitatory synapse. Right, so here we have an excitatory synapse. We have a neurotransmitter that's binding to a non-specific ligand-gated cation channel. Quite a name, huh? Okay, so it's a non-specific cation channel that's ligand-gated. When the neurotransmitter binds, the ion channel opens. Okay, way more sodium rushes in than potassium leaves. So if we actually had a voltmeter in the cell, we would see a depolarizing graded potential, an EPSP. Okay, inhibitory synapses hyperpolarize the cell. And the way they do that is they cause the opening of either potassium channels only. So these are ion channels that only allow potassium through, or chloride channels. Remember, chloride's in higher concentration outside the cell, and it's an anion. And like potassium, its chemical gradient is stronger than its electrical. So if you open chloride channels, it'll come into the cell, and because it's an anion, it'll make the cell more negative. Right? When you open more potassium channels, even more potassium leaves. There's fewer cations inside the cell hyperpolarizes. Right? When you hyperpolarize a cell, you make it harder to reach threshold potential, and therefore less likely to fire an action potential. And so those hyperpolarizing graded potentials that result at inhibitory synapses are called inhibitory postsynaptic potentials, or IPSPs. So here we have a similar figure that we just looked at, but in this case, the neurotransmitter is binding to a ligand-gated potassium channel, right? And this channel is made so that only potassium can move through it. Right? It's very choosy. And so when potassium moves through it, it leaves the cell, leaving fewer cations inside, and it causes hyperpolarization. Right? A hyperpolarizing graded potential. Right? EPSPs and IPSPs are graded potentials, so they can vary in magnitude. They have no refractory period, so that means they can sum. Right? And all the summation can be temporal or spatial. Right? And so you can have multiple synapses firing at once, or the same synapse firing over and over again. Yes?
That was my question.
Yep. Okay. So most postsynaptic cells have multiple synapses with different presynaptic cells. Okay, and it's all about bringing that axon hillock to threshold in specific. So if we look at this figure, here we have a postsynaptic neuron that has synapses with 3 presynaptic cells. Presynaptic cell A, presynaptic cell B, and presynaptic cell C. Okay, A and B are color-coded green because they are excitatory. C is color-coded red because it's an inhibitory synapse. Okay, so here we have temporal summation. We have synapse A firing. Okay, you get influx of sodium. Okay, but then it starts getting kicked out by the sodium-potassium pump. Fires again, a little bit of sodium rushes in, gets kicked back out. Okay, and then that presynaptic cell releases neurotransmitter twice in a row, close enough succession that before all the sodium gets kicked out, even more gets lit in, axon hillock comes to threshold, fire and action potential. Okay, so these two were subthreshold, temporal summation brought it to threshold, fire and action potential. You can also get spatial summation. Okay, so again, A and B are excitatory, C is inhibitory. If A and B fire at the same time, okay, that sodium rushes in, diffuses to the axon hillock, brings it to threshold, fires an action potential. Notice if A and C fire at the same time, C cancels out A, makes it less likely that this neuron is going to fire an action potential. Okay, so C would be inhibitory interneuron that maybe is blocking the signal coming from your inner ear while you're sleeping, right? So even though someone's snoring in the background, you are not consciously aware of that because you had an inhibitory interneuron cancel out that signal, right, before it reached your consciousness. Yes?
So then how is it when like snoring wakes you up then? So is it just like too much stimulant that your inhibitors don't?
Right, you're probably in a lighter phase of your sleep cycle at that point, and at which point a strong enough stimulus could wake you up. Yep, definitely. Okay. Right, any questions about neurons and electrical signaling, synapses, etc.? Right.